Hello, today we're going to be talking about biotechnology. Biotechnology is the use of organisms or their components to make useful products. Okay? Now, with that, genetic engineering is the direct manipulation of genes for practical purposes. And there's a lot of things that we can do in this field. There's a thing called gel electrophoresis. There is inserting genes into other plasmids. There are clones that we could be making. So first let's talk about restriction enzymes. Restriction enzymes are like scissors. They're molecular scalpels that are used to cut DNA at certain points. Now it's a very precise and predictable manner. And what it does is it looks for a palindrome. Okay, now a palindrome is something that is the same going on the first strand forward and on the second strand of DNA going backward. So if we were to do that in the English language, it would be like mom or race car. Okay? And what those restriction enzymes do, they do break the phosphodiester bonds that hold the nucleotides together. So they actually break the DNA bases from the strand off of the nucleotides. Okay? So if you see here, this is where the phosphodiester bond is. Okay? So it will cut those bonds there, and it will make what we call fragments. Now this is saying as a sticky end. This is called a sticky end because these are free bases that will then bind to other nucleotides. There's two different types of restriction enzymes. We have an endonuclease and we have an exonuclease. Endonuclease is going to cut DNA inside or internally, where exo is the outside, so it's going to cut the DNA at the ends. And just to give you some examples of different restriction enzymes, the most typical one is EcoR1. Well, that sounds a little weird, and how that is actually broken down is E will stand for the genus, so Escherichia, coli, so the species coli, R is a particular strain, and the 1 is the first endonuclease isolated. So endonuclease means that it cuts the DNA inside or in the middle. Okay? And then we have BAMH1 and HIN3. So this is just a little bit about EcoR1. All right, now the palindrome. Remember I said that's the place where the DNA is going to cut it. Okay? And it's a very precise and predictable manner because it looks for the symmetrical sequence between the two strands of DNA. So if you look here, DNA is going to be read in this direction for that strand, and it's going to be read in this direction for that strand. So we have GAATTC, and then again GAATTC. So that is a symmetrical sequence. That is a palindrome. Now, where it actually cuts the DNA in the palindrome is called the restriction site. Okay? So if you look here at this, and this is just a little bit of a thing, but when you had the GAATTC, okay, so the GAATTC, it's going to cut it here. And again, this my spacing's off a little bit, so I apologize. That should really be up here. But the G and the C match together, and then this is the sticky end that would result. And then you'd have over here the AATTC and then the G here. Okay, so just to show you a little bit of how it will look, it will cut it there. This is called the restriction site of the palindrome. Okay, so once we take DNA and we cut it up with restriction enzymes, we result in different bands or fragment sizes. So we can visualize the DNA fragments or the band patterns through a process called gel electrophoresis. All right, so we know that DNA is negatively charged. So when we put it in a box that has an electrical current running through it, it's going to travel to the positive end of the box. And that's going to allow the DNA to sort by size. And the polyacrylamide gel is what is going to be, the DNA is going to be loaded in. I'll show you a picture of all this. So as far as the DNA goes, all right, we have it in a micropipette. We're going to actually take it out of here and we're going to load it into these holes or wells. And then this is the power source. So you know that DNA is negative, so when you put the power on, it is going to run to the positive direction. And it will actually separate out the fragments. Now, the bigger banding, the bands that are up top, are going to be much larger than the bands down here, because again, it sorts by size. And this is what an actual one would look like. Again, each of these bands are showing you DNA of a certain fragment size. So this is a reference ladder. So this is showing you that this band is 8 kilobases long. That's 8,000 bases long. That's a lot. 4,000, 3,000. This spot would be 2, and this would be 1,000. Okay. This is showing you of an actual case. The first column is a ladder, and there's actually quite a few ladders in there. A ladder is a known DNA sequence that's been cut by restriction enzymes, and they know exactly how many bases this fragment is, this and this and this. So there's 2 reasons why you would run this ladder. First of all, you want to make sure your equipment's working. It's like a control. And also, you could then measure and figure out exactly how many bases there are. All right, so here's what's important. Here's the victim's DNA. Here's suspect 1, suspect 2. Okay, again, this is a control for the, this was actually the control for the female, and this is what was at the scene, the female. And this was the sperm. You can see that this and this match up together. So it is actually suspect 1. Now, what we just did was what we call RFLP. This is restriction fragment length polymorphisms. Okay, that is taking restriction enzymes and taking the DNA and cutting it into fragments. Again, you get the different bandings when you run it on the gel electrophoresis. Another piece of technology is PCR. PCR is polymerase chain reaction. Now, what this does is this makes a copy of DNA. So this is like a copy machine. And what you do is you place it, the DNA, in a tube, and you add some chemicals or primers into it. And then you put it in a machine, you press start, and the machine will cycle through heating and cooling. Heating and cooling. And as it heats and cools, it is going to generate more copies of DNA. And it can generate 100 billion copies of DNA in just 4 hours. So it's very impressive. So if you look here, if this is the initial DNA, when it goes through PCR once, it makes 2, then it goes through another cycle, 4, and then it keeps multiplying. Okay, so it just keeps copying those molecules. So it is an exponential increase, and it is very effective. Now, why we would want to do that is if you have DNA left at a crime scene, you can't use all the DNA up on testing it. So you need to have extra so the other side, the defense or the prosecution or whatever, needs to test it. Or an outside company needs to validate your results. So you need to have extra copies. It's just like when we make copies of our packets or our tests that we give you. Okay. Short tandem repeats are called STRs. These are locations on the chromosome that contain sequences of bases that repeat. And they repeat between 2 and 5 bases. Okay, this is going to provide greater discrimination, less time, and a sample size. So it is another way to analyze the DNA to figure out the banding. All right. So with that sequence of 2 to 5 bases, okay, the ALU sequence, which is very common in us, is interspersed every 5,000 base pairs in our actual DNA. Okay, so that's pretty impressive as far as that goes. Another example of this would be TCG, TCG, TCG. So 3 bases repeating, TCG repeating. This doesn't require restriction enzymes to analyze. And again, these locations are known. So all we need to do is use a gel electrophoresis, and then you get the band patterns. Okay. So again, you can narrow down the probability of DNA belonging to one person. It's another way to analyze the DNA. The FBI has this database that's called CODIS. It's the DNA database. And it does look at the STR. And these are the 13 core locations for this, for the different chromosomes that we have. Okay? And this is just how you would do it. You would take the sample. All right, and for this one, TH01 has a repeating sequence of AATG. Then we make more copies of it by PCR. We need to run it on a gel electrophoresis to separate out the bands, and then we can figure out how those short tandem repeats migrate through the band and see the actual distance it moves. So the TH01, that STR is AATG. Okay, and then this is what it will look like on a graph. And this would be very helpful in forensics for you to analyze. Okay, so now we're going to be talking about cloning. There's three main types of cloning that we have. All right, cloning is making an exact copy. All right, no variation. So we have reproductive cloning, which is actually making a new animal or organism. So if it's a mammal, we have to put that blastocyst into a uterus for further development. So you take it out and you have your blastocyst. Now that is done by nuclear transplantation. So you take the nucleus of an egg cell or of the zygote, and then you're going to replace it with the nucleus of an adult cell. That's going to cause that cell to divide through mitosis, and that big ball of cells is called the blastocyte. Okay? Therapeutic cloning produces embryonic stem cells for therapeutic treatment. So it's a way to make more cells for you to use for yourself, to feel, to get better therapy or to have a medicine in that. Now, this all cloning, back in 1997, they cloned Dolly the sheep. Since then, they've cloned calves, goats, pigs, rabbits, cats, and the process is what I'll show you in a second. Okay, but again, it's very important that you know that it is an exact copy. There's no variation. Okay, and there are two main types: reproductive or therapeutic. Reproductive produces a new animal. Therapeutic is just particular cells. Now, with this, we can be making recombinant DNA, and I feel like I'm a little out of order, so I apologize. I have cloning coming up in a minute again, so we'll go back to cloning in a second. Recombinant DNA is when you combine genes from different sources. So pretty much DNA from two different sources, and they are merged together. So again, this is a little out of order. I will fix that later. All right, back to gene cloning. All right. First of all, a plasmid is a circular piece of DNA. So you're going to find that, and that's in bacterial cells. You're going to take the plasmid out, and then you're going to find the gene of interest from whatever you're doing. You're going to then insert that DNA of interest into the plasmid, and the bacteria is going to take up that plasmid through transformation, and it will reproduce, forming a clone of those cells with the plasmid that has the gene in it. Okay, so just to show you all of that, okay, again, the plasmid is the circular piece of DNA. You have to isolate that. You take whatever gene of interest you want, okay, and then you actually insert it and you merge it. This is recombinant DNA because it's DNA from two different sources, the source from bacteria and the source from wherever you're taking it from. This gets put back into the cell through transformation, and then it makes multiple copies of that. And then you do have a clone of whatever you've made. So you're going to be doing this in the lab with one of the AP labs with the biotech that you're going to be doing. Again, this is just showing you the same process. All right, and again, recombinant DNA, we already said, just to review that, DNAs from two different sources are combined into the same DNA. And that's just showing you how that's happening. Okay, you cut the DNA, you get those sticky sites, then you can add DNA from another source in there, and then it kind of forms the recombinant DNA. Again, this is DNA from two different sources. These are different products that have been made, okay, from recombinant DNA, and the same thing there. Okay, so I'm going to stop here and you'll watch a second video to finish up the biotechnology. If you have any questions, please let me know.